Well, thank you for a fantastic presentation. Next up, I'll pass over to Ahmet to start his presentation.
Thank you very much, Roshan. So my name is Ahmet Tekin. I'm, as introduced, a senior operational scientist at BioNTech at the site in Marburg, at the Microbial Manufacturing Department, or the Miami project, for establishing a pDNA manufacturing platform. We are one of those customers that Nate and Blaž has mentioned, who had some struggles with our plasmid manufacturing. So we tried out the PATfix and integrated it into our analysis of our steps. To give you a short introduction to who we are, as I mentioned, the microbial manufacturing in Marburg. We are approximately 50 employees. We have different scales for different things we want to accomplish. We have a small scale to manufacture clinical material. We have a large scale for the production of commercial scale plasmids. As far as of now, we only have the COVID-19 vaccine as a commercial product, and our PRIDE project, which is called Process Implementation and Development, in which we try to further develop and make our process better, shortly said. In this presentation, I will go into depth of the alkaline lysis and depth filtration, linearization, and purification, because the transformation, cell banking, and fermentation are not as part of my presentation or as needy. And we don't need the PATfix platform to analyze those steps. Shortly, the PATfix platform helps us to analyze our process. Previously, we only had the UV measurement via our quality control department to check for our content and quality of our product. And because we are a GMP facility, it was a long or a lengthy method, which is GMP validated and takes too long and is not for our benefit. So we reached out to our friends at Bioseparations and Sartorius, and they handed us a solution, the PATfix HPLC, which with a weak anion exchanger, which accomplishes a simple and fast analysis, characterization, calculate our concentrations, and do qualitative and quantitative analysis of our process. In the chromatograph on the right, you see the standard of Sartorius or Bioseparations. In red, you see the open circular and supercoiled standard, and in blue, our self-made linearized pDNA standard, which we use to quantify our product. And in the further detail, which I want to talk about in the alkaline lysis, is what steps or what points in the alkaline lysis we are able to take a closer look at and see how we can make it better. So first off, what we have to see is how can we efficiently get more plasmid out of our yield when we lyse our cells. To see in which points we need to take a closer look at is a convenient mixing equipment or method, because if we rigorously shake our cells during our lysis, we just damage all our plasmid, which is not good. We played around with the suspension buffer volume 10 times, 20 times, and 50 times and checked how we can accomplish a higher yield. Also, we played around with the concentration of sodium hydroxide in our lysis buffer. Our precipitation buffer, we played around a little bit with the final concentration in it, and overall, just our buffer volumes and additions, and checked what the yield said. So we had our different concentrations and yields that we tried to check. And in this, you can see that first off, the first peak is all the contaminants that Blaž was talking about that are shown in the that elute from the column. So we had difficulties in PATfix with our product because the UV signal was too weak because of low concentrations of our plasmid due to high volumes of the buffers. So what we did is we changed our sample loop from 100 microliters to 1,000 microliters, which enabled us to just load more sample and get a longer detection of the signal. Also, what we did is we changed the UV flow cell to get a longer pathway or a stronger signal detection. So in the next step, we take a closer look at our ultra diafiltration step and to check how to not kill our pDNA. Also in this step, we need to be careful and gentle while we're handling our pDNA or when we put our pDNA on our ultrafiltration system, our TFF system, because if we process it too harshly or put it on with too much pressure or too high flow or not the right or optimized parameters, it's easy to clog the filter cassette, or we just produce a gel layer on top of it because that's what the pDNA tends to do if we don't have the right parameters, which also happens to us. So we had to try to find the sweet spot of the different pressures and flows and to not clog our filter, and at the same time not put too much shear stress on our pDNA to essentially break it down. In the next step, how we try to optimize our chromatography is we were thinking about, is it possible to just put the anion exchange directly after the depth filtration and not just go through an ultra diafiltration first? So what you see is on the left directly the anion exchanger or the chromatography after the depth filtration, which shows just a very, very low amount of plasmid in elution. And if we just load it directly after the ultra diafiltration, we saw that the plasmid is in the wash. Our problem was that we probably didn't get our right concentrations. So we started to optimize our buffers a little bit around here. The main problem that we had was that our conductivities of the buffers were not right. So the pDNA and RNA concentrations of the product were too high, and we didn't see a differentiation of it. So when we did our anion exchange, we, of course, also tried to upscale it. On the left, you see only two fractions, which is basically only supercoiled DNA and no RNA. On the right, you see the chromatograph of the anion exchange after ultra diafiltration, and we see that we only have one fraction, and the wash fraction, the open circular supercoiled in RNA. So in the next step, what we try to do is we try to load our column of the chromatography only with a low amount of plasmid. So we try to use it with only 10% of the maximum capacity that the column is able to hold. And what we saw is that it actually worked very well after we increased or decreased DNA concentration, or we didn't put too much, how should I put this? We didn't overload our column with DNA. That's actually what you can see in this slide. This is what happens when we just tried for fun to overload our column with about 200% of its capacity, and we see that we don't get any clear peak. It's just a noisy signal that we get throughout all of this, all of the chromatograph. So another crucial step is the linearization that we try to achieve, because as you know, the mRNA-based vaccinations need to be linearized. or need to have a linearized plasmid as a starting material. We do this via enzymatic linearization of our pDNA. At first, we started with a 24-hour time course of enzymatic restriction. And we optimized this by just taking samples of our linearization process, just taking samples every hour and check how our linearization steps, how our linearization process is shown. In blue, you can see basically time point 0. In red, you see time point 1, and in green, time point 2. So what we can observe is that the supercoiled version basically changes to open circular first, and after that in the linearized version. So we saw, as I said initially, a partial digestion of the DNA, which shows the open circular peak, and afterwards the complete linearization of the plasmid. In the last optimization point, we have our HIC chromatograph, or the HIC chromatography. What we were able to see is we had a double peak in the HIC chromatogram, which is maybe due to secondary interactions and differences in the conformity. Or also what we're able to see is that in an isogradient gradient, it shows no double peaking, but a slight shoulder. So on the left, you see the this so-called twin peaking, what I was talking about. Also, this shows in the PATfix analysis, this twin peaking, when we take a sample out of the HIC chromatograph. So just to wrap this up a little bit, what we were able to achieve with analysis of our process or characterization of our process with the PATfix platform is we were able to see lots of contaminants in our lysis, but also able to see our DNA concentration, and we were able to do a qualitative analysis of the plasmid during the lysis. We were able to see our partial removal of RNA and contaminants, or concentration of DNA, and also that our buffer exchange for chromatography, that it works better if you do it before we load our DNA on the anion exchange chromatography. In the AX, we see itself a complete removal of RNA and the partial separation of the isoforms, because as you know, before we go to linearization, what we want is a supercoiled isoform of the DNA. What we also see is that the linearization is successful after a shorter amount of time, and not the initially started 24 hours of enzymatic restriction. We were able to see that in the HIC, we remove the endotoxin, which, as Blaž was mentioning, has a high similarity to DNA. So we are basically purifying it along with our DNA, but in the HIC, we are able to remove this. And so far, we were able to increase our plasmid yield 8-fold after our first process optimization. And we did this thanks to the PATfix platform, which is able to visualize and easily analyze our samples in a fast, it's basically really fast and easy to analyze our samples without sending it to our GMP validated method QC quality control department, which takes a long time to send us our results back. And with this is basically my end. I will thank you for listening to my presentation, my coworkers who helped me with the presentation and mainly do the PATfix analysis, all of our operational scientists and development team, our production personnel, of course, Sartorius and Bioseparations for helping us with the PATfix platform, and Copic for helping us with the development. And this is it from our side.